Dan Eisenberg from the Department of Anthropology, an associate professor, and he's going to be telling us about paternal effects on offspring telomere length. So thanks for being with us, Dan, and go ahead and take it away.
Okay, thank you for the introduction. And you can see my slides okay and hear me okay? All right, excellent. Okay, so I'm going to be talking about some of my research, which is primarily in telomere biology. And I probably don't need to tell this audience that telomeres are an important biomarker and likely determinant of aspects of aging and senescence. And what I'm going to be focusing on today is some peculiar and I believe important inherited impacts on the telomere lengths that we start out life with. So to remind you, telomeres are repeating sequences of DNA found on the ends of our chromosomes. And each time a cell replicates and the DNA needs to replicate, due to the end replication problem, telomeres tend to get shorter. And eventually, if telomeres get too short, that cell is no longer able to replicate and will either go through cell death or cellular senescence typically. So because telomeres tend to get shorter each time a cell replicates, it shouldn't surprise you that at least in humans, telomere lengths tend to get shorter with age in somatic cells because our cells have been around and replicating for longer. And again, this is important because as telomeres get shorter, cells can't replace themselves as well, and repair to tissue damage is not likely to work as well as things like immune function, which is cell proliferation dependent, probably doesn't work as well. So this leads to the fact that shorter telomeres are associated with poorer health. And at least some of this association is thought to be causal due to cells not being able to replace themselves as readily. So one of the big questions, of course, in telomere biology and in aging biology more generally is why do some people age more rapidly than others, or why do some people have shorter telomere lengths than others? And like for most traits, there's two basic explanations at a very broad level. One is inheritance and the other is environment. And as some of you know, there's lots of interesting studies about environmental impacts on telomeres. I'm not going to really have much time to talk about those, but I'm happy to chat in Q&A or on Slack about that or otherwise. I'm going to be focusing on some weird inherited intergenerational effects on telomere lengths. And as a segue to understand this, I want to focus back to some really basic biology, which you all know, but it's worth a reminder. And we're going to focus on the differences between the birds and the bees, or female versus male inheritance. And I'm particularly focusing on what defines the differences in gametes, which is the number of eggs versus the number of sperm. So women produce all of their estimated 1 million eggs before they're born, whereas men are producing an estimated 50 million sperm per day. Now, if you remember what I've told you about the way telomeres work, this presents a problem for telomeres in our spermatogonial stem cells, that all else equal, we would expect spermatogonial stem cell telomere lengths to be rapidly shortening and for men not to be able to continue to produce sperm very well. Now, of course, our biology has a way around this. And the big way that our biology seems to get around this is this enzyme called telomerase, which helps to rebuild telomeres. And telomerase is not active in most human somatic cells. It's barely detectable most of the time. But one place where it's active at really high levels is in the testes. So this has led to a model where in somatic cells, telomerase activity is mostly off, and telomere lengths correspondingly tend to decline with age. And then since we know telomerase activity is on in the testes, the model has been, well, maybe the first kind of guess was, well, this should help to maintain sperm telomere length with age, just to account for this high rate of cell proliferation and sperm production. Interestingly, this is not what we find. What we find is if we look at sperm telomere length, it apparently increases with age, that older men will have sperm with longer telomere length. And of course, sperm is a particularly important cell. Well, occasionally it's a very important cell, and that occasionally one of these 50 million sperm that's produced per day will form half of the genome of the next generation. And correspondingly, telomere lengths are longer in the children of older fathers. So one of the big questions that this finding led me to contemplate is that whether this paternal age effect, wherein older fathers have offspring with longer telomeres, whether it might be an evolutionary adaptation. And particularly having an older father could signal to a child that it's likely to be in an environment where they're likely to be able to reproduce at a later age themselves. And in such an environment, it might be more adaptive to invest resources in maintaining the body and a long lifespan. Now, one obvious problem with this is that there's a lot of variability that goes into when a man reproduces. I mean, most obviously, birth order is going to make the same man have multiple offspring, in the same man in the same environment have multiple offspring at different ages. So one of the ideas that I explore in my research is that the paternal age effect might persist across multiple generations. And if this were so, then rather than setting somatic maintenance to the age of reproduction of just the father, this could set it in response to the average age of reproduction among multiple generations of recent paternal ancestors. So I'm going to show you a few different results from papers that I've published in the past few years in this talk. The first is going to be testing this first proposition of, does the paternal age effect on offspring telomere length persist across multiple generations? And I'm going to show you a little bit of cross-species variation in paternal age effect and how this might give us some insights into some of the underlying biology that's driving this paternal age effect. And then finally, I'm going to show you some of the most speculative research, which is examining whether factors aside from paternal age may influence offspring telomere length and whether there might be other types of paternal effects, perhaps mediated by the paternal environment. So to start out with part one here, the data I'm going to be showing you from humans, which is going to be most of the data that I'm going to show you, comes from the Cebu Longitudinal Health and Nutrition Survey. I don't really have time to get into the details of it, but we can talk about the study more in Q&A. Suffice it to say, for our purposes, in this most recent study, we had paternal age data and telomere length data for over 3,000 individuals. And then for these individuals, we also, for 2,913 of them, we had telomere length measured, and we had data on the ages of the grandfather at the conception of the mother or father, the respective parent connecting to the grandchild. And so to summarize these results, what we find is consistent with a lot of other research. We find this paternal age effect, and that's not at all surprising. But what we also find is this grandpaternal age effect. So if and this is additive and independent of the paternal age effect. So if the grandfather was older at the conception of the mother or father, the grandchild will have longer telomere lengths regardless of the paternal age. Now, this effect size is about a quarter. The effect size of the grandfather's age on the grandchild's telomere length is roughly a quarter of the paternal age effect. Given the confidence intervals, this is consistent with the semi-conservative nature of DNA inheritance. That is, it could also, the confidence intervals are compatible with the halving with each generation. And this is important because while the effect is considerably smaller and thus less important of the grandfather's age on the grandchild's telomere length, remember we have twice as many grandfathers as we have fathers and twice as many great-grandfathers as we have grandfathers. So while the effect is decreasing, the number of individuals who are influencing this are doubling with each generation and working in the other direction. So to summarize this part two, which is published in this Proc B paper, paternal and grandpaternal ages are associated with longer telomeres in descendants. And the effect size reduces across each generation, but remember the number of grandfathers double with each generation. And this broadly fits with the idea that the multi-generational nature of the paternal age effect could allow it to represent a better signal of the environmental conditions. Now I want to move on to some of this work on examining a little bit of cross-species variation in the paternal age effect. And to understand the motivations for this research, I want to return to considering the mechanisms which may explain sperm telomere length with increases with age. And the first I've already mentioned, which is that there's high levels of testicular telomerase activity, and this could be a chief explanation for why sperm telomere lengths increase with age. But there's also evidence that there may be some selective attrition of spermatogonial stem cells with shorter telomere lengths, or proliferation, specifically symmetrical proliferation of spermatogonial stem cells with longer telomere lengths. And to the extent that this is the case, there may the sperm telomere length increasing with age may be driven by increased rates of sperm production that may be accelerating this process. And rates of sperm production vary across species. And to get at this, I compared chimpanzees and humans. And so to kind of remind you of the motivation here, the paternal age effect is thought to be driven by continual production of sperm with age. And testes size is a big determinant of sperm production rates. And chimps have much larger testes than humans. So the hypothesis is simple here, that if sperm production rate is driving the paternal age effect, there should be a greater paternal age effect in chimps than in humans. And this was supported by our findings that the chimp paternal age effect was approximately 6-fold greater than the human paternal age effect. Again, consistent with this hypothesis that sperm production rate might be a driver of the paternal age effect. Now, finally, I want to move on to this most speculative section, which is whether factors aside from paternal age influence offspring telomere length or influence sperm telomere length. And part of this may just be expected because we have this dynamic nature of sperm production driving changes in sperm telomere length. But there's also some mouse model evidence that leads to clear reasons to expect this. So some experimental work shows that with increased nicotine exposure, there's increased testicular cell apoptosis and decreased testicular telomerase and shortened sperm telomere length. Another study shows that, that an antibiotic that inhibits DNA synthesis and increases oxidative stress decreases sperm density and motility and reduced testicular telomerase. And this was partially restored or reversed by green tea extracts, which have antioxidant properties being administered. And finally, tumor necrosis factor alpha, which, as you probably know, is a pro-inflammatory cytokine, which is produced in response to stressors, has been shown to shorten telomere length in male offspring. So all of this work has been in mice, and for reasons, but mice telomere biology is radically different from human telomere biology. So it's really important to test this in humans. And so returning to the data from the Philippines, I used data from 687 fathers where we had various self-report data and anthropometrics, and used those to predict the telomere length measured at about 22 years of age in 687 of their offspring. And I'm going to show you some of these various variables that we tested momentarily. And with each of these variables, they were motivated by the mouse studies to look at things related to nicotine exposure as well as stressors. And we looked at each of these potential factors along with the individually as well as in interaction with the paternal age at conception to see if there was an interaction effect. So the first most obvious thing we looked at was whether the father reported smoking before the conception of the offspring and whether this predicted the telomere lengths of the offspring. And we found a fat lot of nothing there. And then we used the childhood trauma questionnaire to get at whether stressors, this is particularly looking at linking to that tumor necrosis factor alpha pathway and the idea that stressors might be modifying sperm telomere length somehow. And we found nothing there either. We also used another stressor index where we found nothing. And finally, we used an anthropometric measure of early life adversity, which is knee height. Now I want to pause for a second, explain this weird knee height measure, which you don't see very often. So as you probably know, with poor nutrition and infection or severe stress in early life, we tend to see stunting, that is shorter adult stature. One thing that's less popularly known is that this stunting seems to particularly affect the long bones. And the logic here seems to be that the long bones are more affected to spare the trunk, since the trunk contains these vital organs. So our expectation here is that with poorer conditions, we should see shorter knee heights, and shorter knee heights should thus predict shorter offspring telomere length. And here we found some tentative associations. So to remind you, our expected association is that shorter knee height should predict shorter offspring telomere length. And we found a trend in the opposite direction. And so first I was just puzzled by this and didn't know what to make of it. And then I went back and read the literature a little bit more and found that it turns out that there's some evidence that shorter knee height actually tends to occur with earlier puberty. So in this case, what might be happening is that with earlier puberty, we might be having longer offspring telomere length. So earlier puberty in the father, longer telomere lengths in the offspring. And this could be because if there's an increase in sperm telomere length driven by continual sperm production, but earlier puberty could mean that this sperm production is getting started earlier. So to put this in a graphical way, the way we normally think of the paternal age effect, the age effect on sperm telomere length, is that sperm telomere length increases with adult age. This model is pushing things back to say, well, probably what's happening is that the sperm telomere length is starting to increase at puberty. And if one has earlier puberty, then that increase is starting earlier on. Now, we also examined the interaction between paternal age and knee height. And here we found an interaction where those with shorter knee height had a bigger effect, more positive effect of paternal age on offspring telomere length. And so again, to put this in graphical form, what this interaction effect leads to this slightly altered model where instead of sperm telomere length beginning to just increase at an early age, this suggests that sperm telomere length also increases more rapidly in men who went through puberty earlier. And there may be some sort of programming effect here. Now, of course, these findings are somewhat tentative, especially since we're using such an indirect proxy of pubertal age here, but I find it intriguing that there may be a kind of programming effect that's influencing the telomere lengths that are passed on to the next generation. And let's see, I'll briefly summarize what I've shown you, which is that sperm telomeres increase with age in humans. And this is transmitted across at least 2 generations. And based on the evidence that I've shown you, I think there's good reason to think that the lengthening process may be driven by sperm production rates. And there's some tentative evidence to suggest that earlier beginning of sperm production may cause longer sperm telomeres and a greater rate of increase. And then there's all of this together might be helping to determine the set point of telomeres that we start out life with, which especially accumulating across generations could be highly consequential for aging. And I'll end with the acknowledgements, and if I have time for any questions, I'm happy to take them.
Awesome. Thanks so much, Dan. Yeah, we do have a question for you here in the Slack. Let's see. Were there any sex differences in the telomeres and the offspring of older fathers and grandfathers?
No, I've checked for that, and it's been checked for in some other human studies, and there's no apparent differences. And then the other side of the sex difference that people often want to know about is, well, what about the mother's age, or what about the grandmother's age? And there doesn't seem to be much of any effect. There might be a slight negative effect of maternal age on offspring telomere length. And this makes sense given that we know that the mother produces all of her eggs prenatally, so there's not that same type of dynamic happening in across the life course as we have with men.
Oh, very interesting. Okay, so any other questions for Dan here? Oh, yeah, let's see. So Lena says, did you look at any data where grandfathers reproduced at a later age, but not the fathers? And what was the effect on telomere length?
So what we looked at is I looked at both paternal age and grandpaternal age together in the same model. So especially if I was looking at a paternal grandfather together with the father, both of them were included in the model, and the effect. And so I was modeling them as independent effects, but controlling simultaneously for the other. So I've conceived it. So the way I'm conceiving of it is additively, and that's the way I'd expect the biology to work. And I'm trying to think of. I guess I could kind of try to run some interactions or sub analyses of seeing whether the effects are persist. But I would expect that they would based on the models, although I've not done that exact type of analysis.
All right. Well, very cool, Dan. Yeah, I